talking about carbohydrates in this video. It will be the first molecule that we'll be looking at in our lecture series on macromolecules slash biochemistry. So we need to understand what these molecules are in terms of their structure and their function. You should be able to recognize a diagram of them and tell me what it is they do, where they're found, how they're used by living organisms. So carbohydrates will be the first of the four macromolecules that we're going to be looking at, including carbohydrates, proteins, lipids, and nucleic acid. Four molecules that every living thing uses for some aspect of living, for some aspect of maintaining homeostasis, reproducing, etc. So when you think carbohydrates, think energy mainly. Okay, there's going to be another function, but energy storage, energy conversion, these are the two things that are associated with carbohydrates. You could also think plants when you think carbohydrates. All right, so plants make carbohydrates through a process called photosynthesis. So you've possibly heard of photosynthesis. You've probably, hopefully, noticed that most plants are green. This is because they contain a molecule called chlorophyll. Chlorophyll captures sunlight and the energy from sunlight, and it converts that energy into carbohydrates, molecules that contain that energy. This process is called photosynthesis. And what plants do once they've produced a carbohydrate molecule, such as glucose. Glucose is a sugar, so carbohydrates are sugar. So when plants use photosynthesis to produce sugar, they will then store the sugar molecule as a polymer, as a chain of glucose molecules, usually starch. All right, so here we have a bunch of carrots. Here's the green part where photosynthesis would be taking place, and you probably know that underground the carrot's root, which is the tasty part that we eat, well, they've just stored a lot of sugar there. They've stored a lot of carbohydrates there in the form of starch. That's where most of the energy is. So we eat plants. And even when we're not eating plants, we're probably eating something that ate plants. So all of the energy in a particular ecosystem comes originally from plants and ultimately from the sun. So we consume that and we use the energy from carbohydrates to do whatever it is that we need to live. Okay, including moving is the most obvious one, but thinking, digesting food, many of the chemical reactions that take place in your body require some kind of energy. All of the chemical reactions that take place in your body require some kind of energy. We use carbohydrates to fuel those reactions and those things that we need to do. So what does it look like? We'll build this up a little bit. Okay, but remember last lecture we said that these macromolecules, for the most part, are chains of carbon flanked by particular functional groups. So when you think carbohydrates, I want you to specifically think it's carbon atoms flanked by hydrogen and the hydroxyl functional groups. Okay, they're also going to contain a carbonyl group, which is C double bonded with O. Okay, at the top. So I'll show you this molecule. Typically, okay, these carbohydrates are going to have the chemical formula of CH2O, depending on how many carbons there are in that molecule. Do you know what this molecule is? Okay, if you don't know it, you will know it soon. This is glucose, hexose, 6 carbons flanked by hydrogens and hydroxyl groups. It looks like this. Okay, here's glucose. We say it's a monosaccharide in this form, in all of these forms that we're going to see later. But anyway, a monosaccharide, this is one molecule of glucose, 6 carbon atoms flanked by hydrogens and hydroxyl groups. Pay close attention to that so you don't get mixed up. Here's a hydroxyl group, OH. Here's a hydrogen. Okay, here's the carbonyl group I was telling you about. All right. When you take this molecule, so usually this molecule is not found in a linear form, usually it's found in a ring. So here's a ring of glucose, same atoms, okay, but now it's in a ring. And the hydroxyl group of one end of the ring is going to react with the carbonyl group of the other end of the line, will form a ring. And you could get 2 rings out of this. You could either get an alpha glucose molecule or a beta glucose molecule. You don't need to worry too much about what the differences between these molecules are. It just has to do with where the final hydroxyl group ends up in the ring, is it above the plane on the same side as CH2OH, or is it below the plane? You're going to see that whether or not we're looking at an alpha glucose molecule or beta glucose molecule may change the properties of the molecule. Even when we polymerize it and make a long chain of it, it will change the properties here. So beta glucose tends to be more stable than alpha glucose. Okay, so this is the monosaccharide. This is typically the monosaccharide that most living organisms are going to use, glucose. You've probably heard of it, and you've certainly eaten some of it at some point. Now, I said that this is used as energy. Well, what does that mean? We take the energy stored in the bonds of these glucose molecules, CH, is a very high energy bond, and your body is able to carry out a set of chemical reactions that are going to turn this into a molecule of ATP. Okay, ATP. This stands for adenosine triphosphate, and we will see it in a second. So what is this diagram here that I'm showing you? We're looking at cellular respiration. And there's actually a lot going on here. We won't go through all of it right away. But what does your body do with glucose? What we see here at this end. Well, your cells are going to take up glucose, and they send glucose to a compartment inside of the cell called the mitochondria. We'll talk about the different parts of the cell a few lectures from now, but the mitochondria is the part of the cell that takes glucose and turns it into this molecule of ATP. Why change it into something else? Well, ATP is what the cell uses directly to carry out a particular cellular process. We usually say ATP is the energy currency of the cell. So this word currency, when we use currency in day-to-day language, what are we usually talking about? We usually talk about money, right? So let's say I'm in Canada, I have to go to Europe, right? I'm going to have to take my Canadian dollars and convert them into euros. And the dollar is so bad now, I probably need about 1,000,000 Canadian dollars to buy 1 euro. Okay? The body does the same thing, just more efficiently. We take glucose, and from one molecule of glucose, we can get approximately 25, 29, 32, depending on how you do the calculations, you'll get about 30 molecules of ATP. The body cannot use glucose directly. It uses this ATP molecule for energy. So don't be intimidated by this diagram. We haven't learned about the mitochondria yet, but just long story short, we take a glucose molecule, we go through a series of chemical reactions to make another molecule, which you don't need to know about yet, to make another set of molecules, send them through another set of chemical reactions. All you need to remember for now is the beginning and the end. Glucose goes into mitochondria to make ATP, and ATP is used by the cell for energy. We call this process cellular respiration. Respiration, why? Because it uses oxygen. Okay, so this is kind of giving you an outline of what does it mean to use glucose as energy. Well, we use it to make other high energy molecules in the cell, and that is used as fuel. So going back to the structure now, we have here one molecule of glucose, and here's another molecule of glucose. So by now you've seen the last lecture where we kind of talked generally about macromolecules. Well, what happens when these two monomers, one monomer of glucose, another monomer of glucose, they react, okay, to form a water molecule? Do you remember what we call this reaction? It's forming a water molecule, so we call it a condensation reaction. One thing I don't think was clear enough in the last lecture is these condensation reactions are forming a bond. But what kind of bond is that? Is it a hydrogen bond or a covalent bond? Well, you probably have your notes in front of you, so you see that it's a covalent bond, a very strong bond formed by this sharing of electrons between two molecules. So one monomer plus another monomer, they react, they give you a water molecule, and the resulting molecule is called a disaccharide. Okay, so this word saccharide means sugar. Monosaccharide is one sugar. So glucose is a monosaccharide. When two glucose molecules interact with each other, they form a disaccharide, two molecules of sugar. And the bond that's formed is called a glycosidic linkage. A glycosidic linkage. It's the covalent bond between monosaccharides. So these are two alpha forms of glucose. Here's another reaction that can form a disaccharide. We have beta galactose and beta glucose. And when they react by the condensation reaction, we get something called lactose. Okay, you've probably only heard of lactose if you or someone you know is lactose intolerant. Where do we find it? We find that molecule in milk. Okay, it's um, it's a long story, but some people are not able to digest lactose. They're not able to break down this bond between the two molecules there, and it makes them sick because it accumulates in their body. So just remember those terms. You got a monosaccharide. Whoops, I made a mistake here. Monosaccharide like glucose, one monomer of carbohydrate. A disaccharide, two monosaccharides linked together by a glycosidic linkage. And of course, if you have many of these molecules in a chain, we call that a polysaccharide, a chain. You can kind of see it here, what this is supposed to be representing, or a chain of glucose molecules in a helix. We call this one starch or amylose. This is important. Okay, what kind of stuff am I going to ask you on a test? Well, when we're talking about polysaccharides, okay, a chain of glucose molecules, if it's a chain of alpha glucose molecules, we form alpha glycosidic linkages. Okay, and if they are beta glucose molecules, we're forming beta glycosidic linkages. Okay, and we can make chains out of both of these, but the properties will be different. Okay, they will do different things. So an example or two examples of alpha glucose chains are starch and glycogen. One example of a beta glucose chain is cellulose, and I'll tell you what those are in a second. So alpha glucose chains store energy. What's the one that we've talked about most already? The starch is the first thing that I mentioned in this lecture, one of the first things I mentioned in this lecture. It's an alpha glucose chain. Plants store their glucose molecules, they store their energy as starch. It's a helix, it's a chain of monosaccharides called glucose. So remember that, alpha glucose chain, starch in plants. Anyone know what it's called in animals? How do we store our glucose? Okay, if you're a bodybuilder, any of you guys bodybuilders, do you even lift? We call that glycogen. Some protein drinks and stuff have glycogen. These are branching glucose chains, and they're both used for the same thing. It's just that this is produced, starch is produced in plants, and glycogen is produced in animals. You're going to find glycogen in your liver and your muscles. All right? Did I have to make a muscle gesture here for the video? I guess not, but anyway, in case you don't know what muscles are. Starch and glycogen, animals, alpha, energy. Starch is in plants. Okay, glycogen is in animals. Beta glucose chains, okay, these are held together by many hydrogen bonds, and they're highly packed, and they're difficult to hydrolyze. They're difficult to break down. So when you look on a nutritional label, if you see dietary fiber, well, one of the main components of dietary fiber is this molecule here called cellulose. We can't digest it. This is part of the reason why it's kind of good for you when the bacteria in your digestive tract try to break it down. It creates this foam that helps your lower digestive tract carry away certain toxic molecules like lipids or fats that aren't supposed to linger there too long. Don't worry too much about that. Just remember that cellulose is pretty much the main component of dietary fiber. We're not able to digest it. We can't get energy from it, and it's a structural molecule. There are two other examples of structural molecules of beta glucose chains, chitin and peptidoglycan. This is a word that, I don't know, students sometimes have trouble with. Say it a few times, you'll find it fun. Peptidoglycan. And the main difference between these, there's some structural differences, of course, but what I'd like you to remember is that cellulose is found in plants, chitin is found in fungus and insects. I'm going to change slides here. Okay, fungus, like mushrooms and mold, and insects, well, the structural components of the cells in these organisms is made of a beta polysaccharide called chitin. Remember that word. Okay, and in bacteria, here's some bacteria swimming along, they use something called peptidoglycan. Sorry, look how I drew over that. That was not very considerate. I'm only making it worse, aren't I? I'll write it over again. Peptidoglycan found in the cell walls of bacteria. It's structural. It's difficult to hydrolyze. All right, so that's it for carbohydrates. Next video will be on protein. So what you want to remember, try and study with a buddy, see if you can remember the different forms of this molecule. Okay, what are the two different alpha ones? What are the two different alpha glucose chains? Right? Starch produced in plants, glycogen produced in animals. What are the three different beta glucose chains? Cellulose, chitin, peptidoglycan, okay, etc., etc. And what are these things for? Be able to recognize them on the diagram. All right. See you next lecture. Proteins.